Hey everyone, so I have some pretty exciting science news stories for you today. The first one we're going to be looking at is this really interesting study about cancer cells and their uptake of iron and how this might be exploitable to deliver drugs to cancer cells. So let's dig into this article here from SciTech Daily. Cancer cells' iron addiction may be an Achilles heel. What are these images? Okay, so we see a vertebra. This is a cross-section of something, of the liver. Shows the accumulation of iron in metastatic tumors growing in the spine and liver of patients with pancreatic ductal adenocarcinoma. Okay, so these images are showing somebody who has a certain type of cancer, and this is an iron buildup because of that cancerous tissue. And so this is apparently a problem. Okay, so let's dig into this. What do they say? Researchers at the University of California, San Francisco have discovered that cells carrying the most common mutation found in human cancer accumulate large amounts of ferrous iron, and that this ferro addiction, quote, unquote, can be exploited to specifically deliver powerful anti-cancer drugs without harming normal, healthy cells. And that's a really critical detail. That's a super important distinction for any cancer therapy. Because when you think about something like chemotherapy is very indiscriminate. It'll hurt healthy cells in addition to the cancer cells. And you just hope that more cancer cells are hurt than healthy cells. And this is why there's a huge push in cancer research to find drugs like this or treatments like this that affect cancer cells but don't affect healthy cells. They leave healthy cells alone. So a method like this ticks those boxes. And that's why this is such an impressive discovery and development. The therapeutic strategy, continuing with the article here, described in a study that was published on March 9th, 2022 in the Journal of Experimental Medicine, could be used to treat a wide variety of cancers driven by mutations in the KRAS gene, K-R-A-S. Okay, so let's go to that study real quick, and let's take a look at this paper. So here we have the Journal of Experimental Medicine. Ferrous iron activatable drug conjugate achieves potent MAPK blockade in KRAS-driven tumors. Okay, so I don't have access to the rest of the content, but we can read the abstract here. They say, quote, KRAS mutations drive a quarter of cancer mortality and are mostly undruggable. Okay, so what that means is that whatever these KRAS mutations are, they're mutations that appear in some kind of chemical pathway. We'll get to that in a second. But these mutations seem to exist in a lot of cancers. And as they say here, they drive a quarter of cancer mortality. So out of all the cancer deaths, at least a quarter of them are associated with these KRAS mutations. So that's really significant. And another important thing here is they're undruggable. There's no drugs that can treat cancers that are driven by these KRAS mutations. So that's the problem that they highlight right away. Okay, let's continue. Several inhibitors of the MAPK or MAPK pathway are FDA approved, but poorly tolerated at the doses needed to adequately extinguish RAS, RAF, MAPK signaling in the tumor cell. This is unfortunate with a lot of conditions like cancer or with like psychological conditions, for example. The amount of medicine that you need to treat some of the symptoms or some of the problems, it can often be more than the body can handle safely, and you get these toxic effects. And so that's what they mean here when they say that these drugs are FDA approved, but they're poorly tolerated at the doses needed. So by the time you have a dose that's large enough to actually do something effective to help you with whatever your medical condition is, the side effects are going to be really uncomfortable and probably deleterious in the sense that it might make that particular therapy not even worth it. That's possible. Okay, continuing. We found that oncogenic KRAS signaling induced ferrous iron accumulation early in and throughout mutant KRAS-mediated transformation. Whoa, okay. So KRAS-mediated transformation, this is when the KRAS mutations are causing changes that lead to this tumor production. And so early in that process and throughout that process, they found that there was these oncogenic KRAS signaling processes that induced that iron accumulation, that iron buildup in the cancer cell. So then they say, quote, we converted an FDA-approved MEK inhibitor into a ferrous iron activatable drug conjugate, FEADC as for short, and achieved potent MAPK blockade in tumor cells while sparing normal tissues. There you go. That's what they're looking for. That's awesome. This innovation allowed sustainable, effective treatment of tumor-bearing animals with tumor-selective, like animal models that they were using to test this drug in, with tumor-selective drug activation, producing superior systemic tolerability. Ferrous iron accumulation is an exploitable feature of crass transformation, and FEADCs hold promise for improving the treatment of crass-driven solid tumors. Well, that's pretty freaking cool. Okay, so let's go back to the article here in SciTech Daily. So they explain this in less technical terms. We also get some quotes from the scientists here. Mutations in crass are found in many cancers and are particularly common in pancreatic ductal adenocarcinoma, or PDA. Mutations in crass are found in many cancers and are particularly common in pancreatic ductal adenocarcinoma, PDA, colorectal cancer, acute myeloid leukemia, and lung adenocarcinoma. In total, crass mutations are thought to cause a quarter of all cancer deaths by activating cell signaling pathways that drive cell proliferation and enhance cell survival. So basically, those mutated pathways let the cancer cells replicate faster, more often, and it makes them stronger. They're less likely to be killed by the immune system or by drug treatment. It improves their survival. They can better handle these weird mutations that are making them reproduce like crazy. Continuing, they say, these signaling pathways can be blocked by drugs that inhibit some of the proteins activated by crass. But, in addition to killing cancer cells, these drugs are highly toxic to healthy cells and tissues, limiting their use at doses needed to inhibit signaling in cancer cells. So there you go. There's the dosage problem again. Okay, so Eric A. Collison, MD, a professor in the Department of Medicine at UCSF, said, quote, "For example, inhibitors of the MEK1/2 enzymes have shown clinical benefit, but the approach suffers from dose-limiting toxicities in the eye, skin, gut, and other organs. Clinical experience has shown that sustainable dosing of these inhibitors is often well below FDA-approved dose, severely hampering the dose intensity achievable in the tumor cell and ultimately limiting clinical efficacy." So again, they're reiterating the problem. If you can't give people a big enough dose without causing these horrible side effects, but if you give them a smaller dose so they don't have the side effects, it's not medically useful. And obviously that's a problem. Okay, so now in the article they say here, in the JEM study, first author Honglin Jiang and colleagues develop a method to target MEK inhibitors and potentially other anti-cancer agents to crass-driven tumors without harming other healthy tissues in the body. Super cool. Okay, so here they're getting into it. Adam R. Renslow, a professor in the Department of Pharmaceutical Chemistry at UCSF who led the study, said, quote, "We found that the elevated levels of iron, particularly in its ferrous Fe2+ oxidation state, are driven by oncogenic crass. So we hypothesized that mutant crass-driven PDA tumor cells might be selectively targeted with a ferrous iron activatable drug conjugate, the FEADC." So what are these FEADCs here? They say these are, quote, "inactive versions of drugs that break apart in the presence of iron, releasing the drug's active version." Okay, that's kind of cool. So it's like a Trojan horse, so to speak. So these are drugs that are inactivated, but then when they are absorbed by the target cell, in this case it would be a cancer cell where there's a high presence of iron, the presence of iron there will cause that drug, the whole inactive version, to kind of break apart, and it releases the active version of the drug. So it's like a chemically induced trigger to activate the drug, and that chemical trigger is the environment, that iron-rich environment inside the cancer cell. Super awesome. They say the approach was inspired by anti-malarial drugs like artemisinin that target iron in the parasite as it invades red blood cells and degrades hemoglobin, producing large amounts of free heme iron. All right, well, that's really cool. Okay, so they synthesized this FEADC of the MEK inhibitor cobimetinib. These pharmaceutical names are always crazy. So they found that this drug conjugate, laboratory tests showed that the drug conjugate TRX cobimetinib had little effect on human skin or retinal cells, but it was activated inside crass mutant cancer cells, inhibiting the crass MEK signaling pathway and blocking cell growth. Wow, that is so cool. Okay. The researchers then tested TRX cobimetinib in several different mouse models of crash-driven cancer. Mouse models, that's what I use for my research. There you go. Including PDA and lung adenocarcinoma. In each case, TRX cobimetinib, I am butchering this name, inhibited tumor growth just as well as normal cobimetinib. Whatever. But it caused no detectable damage to other healthy tissues. This lack of toxicity allowed the researchers to combine TRX cobimetinib treatment with other anti-cancer drugs. These combination therapies were even better at inhibiting tumor growth with little side effect on other tissues. Man, this is so intense. Okay, so Collison, we quoted him earlier, says, quote, "In this study, we describe a therapeutic strategy that enables more tolerable and efficacious combination therapies targeting the signaling pathway in crash-driven tumors. The discovery of pharmacologically exploitable ferroaddiction in crash-driven cancer holds promise to improve the treatment of deadly cancers through a practicable and generalizable approach to FE-ADC design, development, and clinical testing." Unquote. Man, that is the coolest stuff. So, to summarize, there's a certain type of cancer that can be propagated by a certain type of mutation. And these mutations, these mutations to this chemical pathway cause the cancer cells, as they replicate and grow, to disproportionately uptake iron. And so now these researchers developed a drug that's inactive until it gets absorbed by a cell that has, this iron-rich environment inside. And that iron-rich environment causes that inactive drug to reconform and break apart and release the active drug, which then affects the cancer cells. It impairs their ability to grow and propagate themselves. And here, coming back to this thing that we saw in the beginning, this is an image of a tumor in a spine, in a vertebral column, and in a liver. And this is the bright yellow you're seeing is the iron buildup. But yeah, this drug is amazing, and it doesn't target healthy tissues because the healthy tissues don't have a sufficient level of iron within them because they're not undergoing these weird mutated chemical pathways where they're absorbing disproportionate amounts of iron. All right, well, that's super cool. Okay, so what do we got next? Our next study, hibernating squirrels can teach astronauts metabolic trait to prevent muscle loss. All right, that title leaves a little bit to be desired. But let's dig into it. What do we got here? So, picture of a cute little squirrel. All right, this is the article from the Science Times. Quote, "During hibernation, bears and ground squirrels stop eating until the spring comes. They rely solely on the fat reserves they've stored up in their bodies so they would not experience muscle loss and reduction in the function of the muscle. But scientists are yet to find out about the mechanism behind this." This article doesn't have the best writing. I'm going to be generous and assume that English isn't the author's first language. But new research led by researchers from Université de Montréal claim to have figured it out, and the implications of its findings could be used for the future of space travel. There we go. That's why this is super interesting. I mean, it is in general, but it has application for space travel, which is, of course, badass. So they studied the so-called 13-lined ground squirrel, commonly found in North America, and confirmed the theory of urea nitrogen salvage that was first theorized in the 1980s. Okay, so we'll get into that in a second. Actually, let's get into it right now. What is urea nitrogen salvage? Okay, so the article explains, a correct balance of nitrogen is essential for life. According to a 2005 paper in Nutrition Research Reviews, mammals' major sources of nitrogen are the amino acids and peptides derived from ingested proteins. So basically, you eat meat or other kinds of proteins, and those proteins are nitrogen-rich. Your body breaks them down, you absorb them, that's how you get nitrogen. At least that's how mammals predominantly do it. And it's probably pretty similar for other vertebrates as well. Now, in mammalian catabolism, its immediate end product is ammonia that is further broken down and results in the formation of urea. But mammals cannot break down urea, which is a byproduct that is found in urine. But urea in the bloodstream can pass through the gastrointestinal tract, where the bacteria utilizes the ammonia as a nitrogen resource and produce amino acids and peptides necessary for growth. These microbial products can be reabsorbed back into the body in a process known as urea nitrogen salvaging, UNS for short. used for synthetic processes. Okay, so that's really cool. So some of these hibernating mammals have evolved a chemical process by which the waste ammonia that's stored in the urea and normally released in the urine, instead, when they're hibernating, this urea can be reabsorbed into the bloodstream, and bacteria in the gastrointestinal tract can use that urea, use the nitrogen and the ammonia, the ammonia and the other nitrogen-containing compounds in this reabsorbed waste material in this urea. It can use that nitrogen to build new amino acids and peptides to maintain muscle mass and to maintain its cells without actually having to hunt for food and eat meat. And that's super important. The fact that animals use this chemical process to help facilitate hibernation without loss of muscle mass, that's a really big discovery. This is like unlocking a key, so to speak, or a key in our understanding of these processes. And now that we have this key, we can apply it. And that's what they talk about next here in this article. How could scientists use urea nitrogen salvage in space travel? The article says, quote, "The urea nitrogen salvage is supposedly the metabolic trick hibernating squirrels do to maintain muscle mass, even after months of hibernation. But how could this be applied to space travel? According to phys.org, researchers posit that the same principle could be applied in helping astronauts minimize their muscle loss problems when in space. Astronauts usually lose muscle mass due to microgravity-induced suppression of protein synthesis, which they try to reduce by intense exercise while deployed in space." So just as a quick aside, when astronauts are in space, in the microgravity of space, all of the stresses that are applied to our body from living here on the surface, from gravity and stuff, they're gone in space. You don't have the constant force of gravity pulling you down, where you have to constantly adjust your posture, for example. When you walk, you have to balance. Your muscles have to coordinate and operate together to keep you upright. And all of this muscular activity, sometimes it's so subtle that you don't even notice it. But it's just your body naturally responding to this gravity environment in which we exist, in which we evolved. But for astronauts who go out into space and they're now in microgravity, all of those background gravitational forces that their body is so used to, that they evolved, that the human body evolved in, and that them individually have gotten used to over their existence on this planet, all of that influence is gone. And so the muscles aren't having all of these small, subtle responses to stresses. And it's there's really, in other words, it's basically just a sudden, almost catastrophic drop in the physical activity of every muscle group. And when that happens for prolonged periods of time, you're going to lose a lot of muscle mass, and that can be really dangerous. Actually, it's bad for your body because dropping muscle mass drastically like that can impact hormone signaling, nitrogen reserves, protein reserves in your own body. It can cause digestive issues, metabolic issues, cardiovascular issues. It's not good for your heart, for example. There's all these issues. And so that's why this research and this discovery of this mechanism of how mammals who hibernate preserve their muscle mass, that's why this is so important. This is it's like a key that's opening a doorway to more exploration in space. Because if we can nail this technology, if we can apply this to humans so that we can potentially hibernate, like astronauts can hibernate in space without losing muscle mass, all of a sudden what becomes feasible, what becomes realistic, is long-term missions to the outer solar system, long-term colonization projects. So perhaps somebody stays for multiple years on a station on the moon or on Mars or somewhere, on an asteroid or an orbital station or something like that. If you can preserve your muscle mass through some genetic modification based off this discovery, that's an important step in this long process of bringing the human species into a true interplanetary, maybe even interstellar civilizational paradigm. Okay, so to further enhance their hypothesis, researchers are continuing to work through a Canadian Space Agency research grant at UDEM. Biologist Matthew Regan from UDEM said that this process might benefit muscle health during spaceflight, if they could prove that there is an overlap of proteins observed during space travel and the ones during hibernation. So basically what that means is if some of the proteins involved in muscle synthesis or stopping muscle synthesis in space are the same proteins, in part or in whole, as used by these hibernating mammals to preserve their muscle mass during hibernation. If there's a lot of overlap, that's going to make it really easy to take this system and apply it to humans, like through some perhaps gene therapy or some kind of a drug treatment or something. However this gets developed and manifested. There's a short little video here, 2 minutes, 17 seconds. We don't have to watch the whole thing, but I just want to see what's going on here. How space exploration affects astronauts' bones. Neat little animation of a spooky skeleton, people running. Okay. Interesting. Okay, so talking about the little microstructures in the bone and how it's supportive, like the struts in a bridge. And that's one thing I didn't mention. Your muscles degrade in the microgravity of space because they're not having stress, applied, and so they're not being activated nearly as much. But also your bones lose a tremendous amount of mass because them too are not experiencing stresses, and so bone growth can stall and bone mass can degrade, and that can lead to a higher risk of fractures, osteoporosis, calcium deficiency, all kinds of stuff like that. What we just saw there looked like it was an animation of some of those internal bone structures thinning and degrading and perhaps causing what's the word I'm looking for? Mechanical compromise. So the astronauts have to work out constantly and pretty intensively to keep their muscle mass. But if you have a technology like this, inspired by the three-lined nesting ground squirrel, then perhaps they don't have to work out so hard. Perhaps spaceflight can be its threat to our muscle and bone health can be mitigated somewhat with a advanced genetic therapy. So let's go look here at this is the article, of course, the paper that was published in Science. Nitrogen recycling via gut symbionts increases in ground squirrels over the hibernating season. Okay, so don't have full access, but we can read the statement of importance or statement of significance, and then the abstract. Okay, let's start with the abstract. Hibernation is a mammalian strategy that uses metabolic plasticity to reduce energy demands and enable long-term fasting. Fasting mitigates winter food scarcity, but eliminates Fasting mitigates winter food scarcity, but eliminates dietary nitrogen, jeopardizing body protein balance. Here, we reveal gut microbiome-mediated urea nitrogen recycling in hibernating 13-lined ground squirrels, Ictodomus tredecimal lineatus. Ureolytic gut microbes incorporate urea nitrogen into metabolites that are absorbed by the host, with the nitrogen reincorporated into the squirrel's protein pool. Urea nitrogen recycling is greatest after prolonged fasting in late winter, when urea transporter abundance in gut tissue and urease gene abundance in the microbiome are highest. These results reveal a functional role for the gut microbiome during hibernation and suggest mechanisms by which urea nitrogen recycling may contribute to protein balance in other monogastric animals. Okay, so that is extraordinarily fascinating. One thing I want to point out here is, so they say, after prolonged fasting in late winter, so this is near the end of the hibernation period, after the animal has been hibernating, a while, its stores of energy that it's built up over the summer are probably depleted or getting close to being depleted. And when it's in that context, when the animal's body is, sensing that it's in this state of, okay, our reserves are running low or they might be out, we can the body starts upregulating some of these other processes that will begin to recycle this urea nitrogen. And so you see these urea transporters, which would be like proteins in gut cell membranes to help move urea across the membrane, as well as urease genes. So these are genes that produce urease enzymes or enzymes that break down urea. The transporters and the urease enzymes are greatest in abundance in the microbiome at this prolonged fasting stage, in the late winter stage. So that's really cool. These are these processes that get ticked up as the animal is experiencing increasing resource stress. And when these transporters and these genes, when they all get in place and they're all activated, it helps the animal preserve its muscle as it hibernates. And so when spring comes around and it gets out of hibernation, it has the muscle mass and the energy where it can just get up, shake itself off, wake up over a period of hours, days, or maybe even a couple weeks, and get right back to it, start hunting again, and getting some fresh protein, some fresh nitrogen. Okay, so now this statement of significance, just the authors explaining this a bit more. Hibernation has evolved to remove animals from seasonal periods that are especially challenging for survival. Despite this protective feature, hibernation poses its own challenges because of the extensive fasting period. One particularly challenging aspect is the lack of dietary nitrogen, which can lead to protein imbalance. Regan et al. Looked at those are the authors. Looked at gut microbiome activity in hibernating 13-line ground squirrels and found that symbionts recycled nitrogen from urea into their own metabolites, which were then incorporated by the squirrels, allowing them to maintain protein balance. These results reveal the importance of the gut microbiome to hibernation and suggest that gut microbes could play such a role in other species. Okay, so now that highlights a point that I've kind of understated so far, is that this chemical process that's been evolved, this nitrogen recycling, it's not purely in the mammal's body. It's not the mammal with its own mammal genes doing this all on its own. The actual recycling of the urea, of the urea nitrogen, it goes through the gut microbiome first. So this is a symbiotic process. It requires, the necessary gut microbes to first absorb that urea and break it down into some more bioaccessible nitrogen. And then that nitrogen, those more bioaccessible compounds, are released back into the bloodstream, into the gut, and the animal can use them. They're added back to the animal's what they call protein pool. And so that means that if we were to use this finding and apply it to humans, it might take the form of a genetic therapy, but it's quite possible that it might actually take the form of a microbiome transplant, like a fecal transplant or something like that. And I mean, this is just a really cool example of how biological science is just mind-blowing, how crazy it is. So we could theoretically use a fecal sample with microbes taken from the gut of a hibernating ground squirrel. You take that pill, you introduce those microbes to your gut flora, your gut microbiome, and all of a sudden you might be able to endure long stretches in space as an astronaut without suffering muscle loss or potentially even bone loss. We have to study a bit more to see how widespread the effects of these processes are. But this is just super cool and super exciting. This is a great science news story for how just some random chemical and some random animal can have profound impacts on human civilization. It's super cool stuff. Okay, now let's go to our third and final news story for the day. This one's a tad terrifying, if I do say so myself. An AI experiment generated 40,000 hypothetical bioweapons in just 6 hours. Okay, so we'll get to this article, but first I want to dig into the study. This is a preview of subscription content. I don't have the subscription, but let's just read what we can see here. Dual use of artificial intelligence-powered drug discovery. An international security conference explored how artificial AI technologies for drug discovery could be misused for de novo design of biochemical weapons. A thought experiment evolved into a computational proof. So that's basically what this paper is. It was a thought experiment inspired by some work at the security conference that was developed by the authors into a computational proof. So we can't read this whole paragraph here, but it begins. The Swiss Federal Institute for NBC, Nuclear, Biological, and Chemical Protection, SPIE's laboratory, convenes the Convergence Conferences series set up by the Swiss government to identify developments in chemistry, biology, and enabling technologies that may have implications for the chemical and biological weapons conventions. Meeting every 2 years, the conferences bring together an international group of scientific and disarmament experts to explore the current state of the art in the chemical and biological fields and their trajectories, to think through potential security implications and to consider how these blah, blah, blah. I can't read beyond that. It is covered. Got to pay $29.99 a month to get your subscription to that. Not going to do that today. But let's go back to the article. So this is in Science Alert. Okay, so at the top of the article here, quote, "The cutting-edge number crunching capabilities of artificial intelligence means that AI systems are able to spot diseases early, manage chemical reactions, and explain some of the mysteries of the universe. But there's a downside to this incredible and virtually limitless artificial brain power. New research emphasizes how easily AI models can be trained for malicious purposes as well as good, specifically in this case to imagine the designs for hypothetical bioweapon agents. A trial run with an existing AI identified 40,000 such bioweapon chemicals in the space of only 6 hours. In other words, while AI can be incredibly powerful and much, much faster than humans when it comes to spotting chemical combinations and drug compounds to improve our health, the same power can be used to dream up potentially very dangerous and deadly substances." Okay, so there's your brief introduction there. If you've watched my science news segments, in the past I've covered a couple science news stories about like AlphaFold and some of these other AIs that have been developed to address long-standing problems in biology. Like AlphaFold, for example, sought to resolve the folding issue in protein research. Namely, if you start out with just the amino acid sequence of a protein, can you go from that information to determine what the final three-dimensional folded structure of the active functional protein looks like? That's been an extremely difficult problem that was expected to go unsolved for 50 years. But with the development of these new AI technologies, these advancements in understanding protein folding and other related issues that we would use AI for in the medical field, so like radiology, diagnostics, that kind of stuff. AI has allowed for these rapid, unprecedented advancements in our knowledge and understanding, and our ability to do these things, to diagnose cancers early, or to predict the structure of a complex protein. And in that sense, we could modify proteins at the amino acid sequence level and have that produce the intended results or features that we want in the final protein. And that is an extraordinarily powerful technology. And again, we didn't think we'd be able to develop this for like 50-something years. But these new AI technologies are making it something that's perhaps achievable in the next few years, the next decade. And so while that's all super awesome, that we're solving the protein folding issue, that we're developing new diagnostic technologies like radiology AIs and stuff, there's a dark side to this. The very same technologies, the very same AI programs can also be used to do bad things, like develop bioweapons. And in this case, just this brief trial run where this hasn't even really been studied too intensively. They're not using like some sophisticated bioweapon genesis program that was developed by some malicious actor. This is just something they were putzing around with, and they were able to create 40,000 bioweapon chemicals in 6 hours. I mean, it's terrifying because you think about how widespread these AI technologies are today, and how they're only becoming more and more widespread. More and more people are getting access to them. More and more people are going to get abilities like this. And, access to chemistry labs is pretty easy for most people. It's publicly accessible resources in most countries. And so the potential for, a dirty bomb, like a dirty bioweapon to be used, developed by some amateur in a lab using some publicly accessible AI program like this, it's suddenly so much more feasible, so much more realistic. And it's really, really terrifying because with that feasibility comes a certain level of unpredictability, of potential chaos and destruction. The researchers, they explore all of these issues in their commentaries and their interviews and stuff. So they're quoted in the article as saying, "We've spent decades using computers and AI to improve human health, not to degrade it. We were naive in thinking about the potential misuse of our trade, as our aim had always been to avoid molecular features that could interfere with the many different classes of proteins essential to human life." Unquote. So they put an AI system called Megasyn to work. Not in its normal mode of operation, which is to detect toxicity in molecules in order to avoid them, but to do the opposite. In the experiment, the toxic molecules were kept rather than eliminated. And what's more, the model was also trained to put these molecules together in combinations, which is how so many hypothetical bioweapons were generated in so short a time. In particular, the researchers trained the AI with molecules in databases of drug-like molecules, instructing that they be like something similar to the potent nerve agent VX. As it turned out, a lot of the generated compounds were even more toxic than VX, which is a chemical nerve agent. As a result, the authors behind the new study are keeping some of the details of their research secret and have seriously debated whether or not to make these results public at all. I mean, there you go. That's one of the most real examples of the security threat that something like this poses. That the researchers aren't even willing to publish all of their results because some of the information they uncovered, some of the techniques they may have developed, are just that dangerous. So they say, quote, "By inverting the use of our machine learning models, we had transformed our innocuous generative model from a helpful tool of medicine to a generator of likely deadly molecules." Unquote. And they say it doesn't take much to flip the switch from good AI to bad AI. While none of the listed bioweapons were actually explored or put together in a lab, the researchers say their experiment serves as a warning of the dangers of artificial intelligence, and it's a warning that humanity would do well to heed. While some expertise is required to do what the team did here, a lot of the process is relatively straightforward and uses publicly available tools. The researchers are now calling for a, quote, "fresh look," unquote, at how artificial intelligence systems can potentially be used for malevolent purposes. They say greater awareness, stronger guidelines, and tighter regulation in the research community could help us to avoid the perils of where these AI capabilities might otherwise lead. Our proof of concept thus highlights how a non-human autonomous creator of deadly chemical weapon is entirely feasible, the researchers explain. Without being overly alarmist, this should serve as a wake-up call for our colleagues in the AI and drug discovery community. Unquote. Well, damn, that's terrifying. That's a really scary way to start the week. But there you go. There's some of your awesome biologic science news stories for the time being. I'll try and come back at you in the near future with some more. But yeah, this last story, this is going to keep me up at night, I think. Because it's really cool that AI is being used to, diagnose cancers, solve protein folding problems, and all sorts of other stuff. But this is a really serious risk. This is AI being used to create bioweapons like this in the manner and the method that they described in their paper here. This is upturning a paradigm in a way that's analogous to the use of drones in warfare. To really get this point across, how unsettling and creepy and how profoundly significant a change like this could be, to keep using that metaphor or the analogy of the drones in warfare, just look up that short film called Slaughterbots or whatever it's called. It's like a short film, a sci-fi film about the use of drones equipped with bombs and AI facial recognition software. So they release the drones. They identify the faces of enemies or undesirable members of the population based on perhaps data taken from social media profiles. The drone will fly right up to their head and detonate the ordnance and killing them. And the thing that's creepy about that is, while it's technically not real right now, all that technology is completely feasible. It's all completely realistic in the sense that if somebody wanted to do that, everything necessary to do that is out there. It's all available. Somebody could put it together and build those Slaughterbots tomorrow. And in much the same way, just like is explained in this research here, some random amateur asshole could take one of these AI, develop a bioweapon, access a public biochemistry lab, synthesize some stuff, mail it off to a politician, drop it in a public water supply or something. The potential for destruction is staggering. Really, it's unprecedented. This is what really brings meaning to the phrase, it's a brave new world, because these are modern threats from modern technologies that we are going to have to really have some bravery to address and control before it gets out of control and the world suffers for it. All right, well, on that note, there you go. There's your biologic science news. See you later, guys.